Well, good evening, everybody, and welcome to the Royal Society, especially if this is your first visit to the Society. I'm Jean Thomas, and I'm the Biological Secretary, and it's my pleasure to introduce the Croonian lecturer this evening. But before I do that, could I ask you to switch off your mobile phones, please, because the lecture is being webcast live and also recorded for the Royal Society archives. Okay, thank you very much. The Croonian lecture is the Society's premier lecture in the biological sciences. It's held in honour of Dr. Croon, one of the original members of the Society, which was founded in 1660. He died over 300 years ago in 1684. His will provided the means for establishing this lecture, and the bequest specifically was for, and I quote, the support of a lecture and illustrative experiment for the advancement of natural knowledge on local motion, or, conditionally, of such other subjects as, in the opinion of the President for the time being, should be most useful in promoting the objects for which the Royal Society was instituted. I think that's where we are this evening. The 2011 Croonian Lecture has been awarded to Professor John Ellis, Fellow of the Royal Society, for his pioneering contributions to biochemistry, molecular biology, and also plant sciences. Professor Ellis took his BSc in botany and his PhD in biochemistry from King's College in London. He then worked at Imperial, Oxford, Aberdeen, for a short time as a visiting professor in Toronto, and in 1970 he moved to Warwick to the newly created Department of Biological Sciences. He obviously liked it there because he's been there ever since and became Emeritus Professor in 1996. John Ellis is known worldwide for his work on protein chaperones and indeed for establishing the general concept of protein chaperone function, which has wide implications in health and disease, and not only for plants but for all organisms. And I won't tell his story. I'm sure we'll hear more about that from him shortly. John has also recently written a book published by Springer in 2010, How Science Works, Evolution. So if you want to know, read that book. Very accessible. John Ellis has been the recipient of a number of awards, perhaps most noticeably the Gairdner International Award in 2004, and of course the other important award, the Croonian Prize Lecture from the Royal Society, of which he's a very worthy recipient, and this is why we're here this evening. So, ladies and gentlemen, it's my very great pleasure to introduce the 2011 Croonian lecturer, Professor John Ellis, to speak on the subject Molecular Chaperones, How Cells Stop Proteins from Misbehaving. John.
Good evening, everyone. I should like to start by thanking Jean Thomas for her kind remarks and the Royal Society for honouring me with the Croonian Lecturer Prize. What I'm going to do in this talk is to take you on a journey. It's a personal journey, and it starts in 1970 when I started to study the synthesis of the enzyme Rubisco in the chloroplast-supplying plants. But it carries on to the present day when I'm invited to talk to neurobiologists about aspects of human neurodegenerative disease. Now, if you look at the screen, you'll see the image on the left-hand side is a section through a chloroplast isolated from a spinach leaf. The image on the right-hand side is a section through a human brain. And you may well wonder what connects these apparently disparate topics. Now, the answer is that they share the same problem. It's a biochemical problem involving proteins, and it's a universal problem. It's found in all cells, bacteria, plants, and animals. This problem has a name. It's called protein aggregation. Now, protein aggregation is defined as the formation of non-functional complexes between one or more identical or very similar protein chains. The aggregates can be small as a dimer, consisting of just two protein chains, or they can be large enough to be visible in the light microscope. They can be large enough to consist of thousands of chains which are insoluble. The smaller aggregates in particular are often toxic to cells, including neurons. Now, amyloid plaques are a type of insoluble aggregate found in the brains of people suffering from neurodegenerative disease. And what you see on the screen on the right are amyloid plaques found in the brain of a person who has died of Alzheimer's disease. Now, these plaques are called amyloid because their discoverer thought they were made of starch. This error was soon corrected. They're actually made of protein, but the name has persisted. Now, it was an American called Sam Wildman who discovered in 1971 that the chloroplast enzyme Rubisco also suffers from this problem. It's prone to form insoluble, non-functional complexes. And it was the serendipitous discovery I made in 1980 about how chloroplasts cope with this problem that led to the formulation of a new general concept in biochemistry, the concept of the chaperone function. Now, the basic idea is that many macromolecules run the risk of interacting incorrectly because of the complexity of their surfaces. It's the surface shapes of macromolecules that determine their properties. But these surface shapes change with time. They change when the molecules are being synthesized, when they're being assembled, when they're carrying out their normal biological function, when they're being degraded, and when they're being subjected to environmental stress, such as high temperature. I've indicated some of the stages on this slide. At each stage, there is a probability that these surfaces may undergo incorrect interactions. Now, the term incorrect is defined as meaning that the macromolecules no longer carry out the function they were selected for during evolution, but may in addition have acquired toxic functions. In other words, incorrect interactions result not only in the loss of normal function, but often in a gain of toxic function. The best studied incorrect interactions involve proteins, but the same general argument also applies to other macromolecules, especially RNA. RNA is in fact more prone to incorrect interactions than our proteins, although as yet there is no evidence that such interactions cause human disease. Now, what do cells do about this? Well, they fight this problem. They combat this problem with the aid of a large range of different proteins, all of which act as molecular chaperones. So called because they reduce the probability that other macromolecules will misbehave. Now, why do we call them chaperones? Well, of course, we use it by analogy with a human chaperone. What does a human chaperone do? Well, its role is to prevent inappropriate interaction between pairs of people, as you see illustrated on this cartoon, which was kindly drawn for me by Kathy Raphael. The human chaperone on the right is trying to reduce the probability that the two people on the couch will interact incorrectly. Now, in animals, when the chaperone function fails, as it often does with age, the result is neurodegenerative disease. What I show here are the amyloid plaques that are characteristic of 5 of the most important neurodegenerative diseases. In the UK alone, there are over 1 million people suffering from this type of disease. There are, in addition, another 40 or so diseases that are associated with the accumulation of protein aggregates, but these diseases are less common. Now, in plants, there is also a potential problem due to protein aggregation, and this is in the case of the chloroplast enzyme Rubisco as a particular example. Now, Rubisco sounds like the name of a breakfast cereal, but it is in fact an appropriate name because without this enzyme there would be no breakfast, or indeed food of any kind. Rubisco is arguably the world's most important enzyme because it catalyzes the reaction in photosynthesis which brings carbon dioxide into organic combination. It produces a compound called phosphoglycerate, so the actual reaction catalyzed with Rubisco is this one, ribulose bisphosphate plus CO2, to give phosphoglycerate. And it's from phosphoglycerate that ultimately virtually all the compounds in the biosphere are derived. It's derived by what is called the Calvin cycle, which is driven by these two molecules, ATP and NADP, and these are continually supplied by the energy absorbed by chlorophyll. The whole thing is driven, of course, by sunlight. This makes Rubisco, of course, of central importance. But despite this, it is also the world's most inefficient enzyme. There are 3 problems with Rubisco. The turnover number of an enzyme is the number of molecules acted on per second per molecule. And for most enzymes, this number is in the hundreds to the thousands, or even the millions. But Rubisco has a pathetically low turnover number, somewhere between 3 and 10, depending on the species. That means one molecule of Rubisco fixes only 3 to 10 molecules of CO2 per second. Now, what does this mean? It means that the plants have to make very large amounts of this enzyme. In fact, if you grind up a leaf, you find that up to half the total soluble protein in the leaf consists of just this one enzyme. This makes Rubisco the world's most abundant protein. The second problem with Rubisco is that it binds carbon dioxide weakly at the current level of gas in the atmosphere. This is why some growers augment the concentration of carbon dioxide in their greenhouses so they get increased yields. But it's the third problem that's the most serious. Rubisco also reacts with atmospheric oxygen. This, of course, does not bring carbon into organic combination. It produces another compound called phosphoglycolate. And this phosphoglycolate is the substrate for a metabolic pathway peculiar to plants called photorespiration, which results in CO2 loss from the plant. So we have a very curious paradox here. The same enzyme that brings carbon dioxide into organic combination also catalyzes a reaction which feeds a pathway that leads to CO2 loss. Now, why is it like this? Well, none of this mattered when Rubisco first evolved, because at that time there was no oxygen in the atmosphere, and the CO2 concentration in the atmosphere was much higher than it is now. So Rubisco is a superb example of unintelligent design. If you knew how the world was going to change, you wouldn't have chosen it. The challenge, therefore, is to try and improve this enzyme with genetic engineering. But this is where you run into the protein aggregation problem. Rubisco is a large oligomeric enzyme. It's got a central core of 8 large subunits, surrounded by 8 small subunits. The large subunits are made inside the chloroplast. But during this process, they have a very strong tendency to form insoluble aggregates with one another. And the sheer complexity of the chaperone machinery that deals with this problem has to this day defeated all attempts to improve the properties of this enzyme and thereby improve agricultural productivity. We can't even express the chloroplast enzyme today, with all we know, in E. coli, the organism that's commonly used to engineer proteins. Such is the severity of this problem that I'm currently involved in an ambitious synthetic biology project that aims to replace chloroplast Rubisco with a different and more efficient carboxylation system derived from bacteria. But that's another lecture. Now, before I discuss the evidence for these conclusions about the problems of protein aggregation and how chaperones go about combating it, I've been advised that, particularly at my age, I'm 76, please say I don't look it, it may be of general interest in this sort of lecture to relate some personal experiences about my life in science. The first thing I've learned is it's important to find yourself a working environment that's mentally stimulating. I found this actually to be crucial in my early career. I was fortunate in having 2 inspiring teachers, both of whom I'm happy to say are in the audience. Brian Youngs sparked my interest in biology at grammar school, while Charles Pasternak encouraged me to learn biochemistry during a postdoctoral fellowship in the biochemistry department of Oxford in the 1960s. This shows Charles Pasternak escorting myself and my bride into the taxi that took us on our honeymoon. That's Charles Pasternak, and that's me, and that's my wife. You can tell this was a long time ago because at that time you'll notice I had a full head of hair and a waist. Now, a year later, our daughter arrived as my fellowship in Oxford ended, so I needed a job, and I accepted one in the Department of Botany in the University of Aberdeen. Now, why did I do that? Well, I did it because it looked rather nice. The department was in new buildings, located inside the Botanic Garden. I was given a large new office, a huge lab, a technician, a Spinco ultracentrifuge all to myself, and access to the electron microscope. So my lab occupied the entire lower floor, that wing, and this was my office. So physically, the environment was very attractive. But I soon discovered to my dismay that intellectually this department was a desert, resting quietly in the 19th century. There were some members of staff who actually thought that plants were not made out of chemicals. There were others who did admit this, but said the idea you could find out anything important about how plants work by grinding them up in a blender and studying the bits was clearly absurd. Now, I found this attitude profoundly disappointing. You must remember that this was the 1960s. And during that decade, most of the basic concepts of molecular biology that we use today were being laid down. The genetic code, the central dogma, and the principle of Alice theory. To my mind, the single most important principle in biochemistry. There have been huge developments in biochemistry since then, of course, but to my mind, these advances have been largely technical rather than conceptual. So the 60s were an exciting time for a young biologist interested in new ideas. Now, it so happens that the University of Aberdeen also has a department of biochemistry. This was about a mile away from botany. So what I found myself doing was spending more and more time in biochemistry, talking to the staff, attending seminars. Now, in the winter in Aberdeen, you have to be really keen to do this. You have to get up in time to be at the Saturday Journal Club at 9 a.m, and when you come out at 10 a.m, it's still dark. It was my wife who had the good sense to say to me in 1968 that there was a lectureship being advertised in the biochemistry department, so why didn't I apply for it? And this turned out to be the second most important thing my wife has ever said to me. To tell a long story short, I moved from being a lecturer in the botany department to being a lecturer in the biochemistry department, which is possibly a unique move. Now, the physical state of the biochemistry department is very poor compared to botany. In fact, these days you wouldn't be allowed to work in it because of the fire hazard. My office was a closed-off corridor. You could touch the walls on each side just by stretching your hands out sideways. One postdoc I met there was John Scale. Now, Sir John Scale recently retired from being director of the Medical Research Institute at Mill Hill. He worked in the laboratory of the virologist and cell biologist Derek Burke. And the lab facilities were so restricted that John Scale's workbench was actually the top of a deep freeze. So the physical environment was poor compared to botany, but the mental environment was much better. Now, my move to biochemistry proved to be serendipitous because 1 year later, Derek Burke was appointed head of the newly appointed Department of Biological Sciences at Warwick, and he invited me to apply for the second post. So I moved to Warwick in 1970, and thus started the most productive part of my life in science. And I'd like to thank Derek Burke for his sterling support in this critical period of my life. The second take-home message is to hope that when you do an experiment, you get a result you did not foresee. Now, when you carry out an experiment, there's usually an expectation that the result will fall within a range of predictable outcomes. And when this happens, it's natural to feel pleased. But in my view, this is a response. This response is a mistake. What you should be hoping for is an unexpected result, something you didn't foresee, one you had not anticipated. Because with persistence and luck, and you do need both, you may have come across something new and of wide significance. And this turned out to be the case when I studied the synthesis of Rubisco, as I'll explain in a minute. The last take-home message refers to the value of Occam's razor. Now, in my view, Occam's razor is the single most important principle that distinguishes science from other ways of interpreting the world, but you do have to use it with caution. Let me remind you about Occam's razor. William of Ockham was a Franciscan philosopher who lived in the village of Oakham in the county of Surrey. There's actually a pub in the village named after him. Now, he tackled the problem that for any given body of evidence, you can always postulate several different possible explanations, varying in complexity. And William argued that in this situation, the best way to proceed is to prefer the simplest explanation, the explanation that makes the least number of assumptions. The word razor is used to mean that unnecessary assumptions are shaved away. Now, it's very important to grasp that Occam's razor does not say that you should prefer the simplest hypothesis because it is more likely to be correct. This is a very common misconception because there is no a priori reason why nature should be simple. Biology certainly isn't simple. What Occam's razor says is that you should prefer the simplest hypothesis because it is the best way to proceed. In other words, he's defining a method. And it's an essential part of the method that all scientists use. Now, I can't overemphasize the importance of Occam's razor to the practice of science. If you abandon this principle, you might as well believe any interpretation of the world that you find comforting and appealing, and many people do just that. I suggest that it's because of Occam's razor that the majority of leading scientists today, according to surveys, are not supernaturalists. Because postulating supernatural agents clearly requires more assumptions than does the naturalistic view. Naturalism, you'll recall, is a philosophical view that natural events have only natural causes. This is an assumption that scientists, including religious scientists, necessarily make when they practice their science. However, although Occam's razor is vital to the practice of science, it's not infallible. It can mislead you. It can mislead you because the actual explanation of a phenomenon may not have occurred to you. It may not be amongst the range of possible explanations you've thought of. This is the problem that Donald Rumsfeld famously called the problem of the unknown unknowns. Now, I also ran into this problem when studying the synthesis of Rubisco. Now, Occam's razor brings me to the advertising slot in this lecture. This slide is taken from a book that I published last year, as Jean Thomas mentioned, by Springer. It's based on a course I give to the students at Warwick on the philosophy of science and how it differs from other ways of interpreting the world. And it uses evolution as the example. Now, this book has received some favorable reviews, so what I've done with the the agreement of the publisher is arrange for 100 copies to be made available just outside the lecture hall. Now, on the right, I've listed the questions I discuss in this book, so if you find these questions of interest, you might like to exchange the voucher you've been given on arrival for a copy of the book when you leave. Now, let me emphasize, this book is free. You do not have to pay for it, but you might prefer to read it online. You can find websites that have this book by putting the title into Google. Now, what I'm going to do in the remainder of this talk is to discuss some of the evidence that led to the idea of the chaperone function. And what I first need to remind you about is one of the basic principles of biochemistry, the principle of protein self-assembly. If we ask what determines how proteins fold into their functional conformations, the answer is on this slide, which describes the classic refolding experiment of Amundsen, first carried out in the 1950s, and which earned him a Nobel Prize in chemistry in 1972. What you do in this experiment is to take a pure native protein, a simple buffer, you expose it to an agent which destabilizes the non-covalent interactions that hold the protein into its functional conformation. So the protein unfolds, it loses its activity. And what Amundsen found, and in fact he found it serendipitously, I might say, that if you reduce the denaturing agent, in many cases the protein will refold spontaneously and regain its biological activity. And it will do all this on its own. You don't have to add anything. Now, many repetitions of this type of experiment show that many proteins will, in fact, refold correctly in the absence of either other macromolecules or even a source of energy. Now, I was explaining that Amundsen's experiments, original experiments, were done with monomeric proteins, proteins consisting of just one chain. But most proteins, in fact, are more complex than that and consist of more than one chain. And so what happens is that after the chains are folded up, they're assembled together. And I just showed this slide to make the distinction between protein folding and protein assembly. And in vitro experiments of the classic Amundsen type show that both these processes will occur spontaneously in the test tube. Now, if we apply Occam's razor to these interpretations, the simplest assumption that proteins also fold and assemble spontaneously within the cell. And in fact, this is what it says in the textbooks for many years. But observations made in the 1980s challenged this view. 2 reports appeared that protein chains newly synthesized inside the cell, in fact, bind transiently to pre-existing proteins before they fold. And the first report was for the enzyme Rubisco, which was made in my laboratory at Warwick in 1980. Now, in the 1970s, my lab was studying the biogenesis of plant chloroplasts, trying to determine what genes they contain and what proteins they make. Chloroplasts are interesting because, like mitochondria, they have a complete genetic system additional to one in the nucleus. But unlike mitochondria, they contain very large numbers of ribosomes. In fact, about half the ribosomes in a leaf reside within a chloroplast. So we ask the question, well, why are so many required? Are they making a high proportion of the several thousand chloroplast proteins, or are they only making some of them, and if so, which? So we tackled this question by incubating chloroplasts from expanding leaves of young pea plants and incubating them with light as a source of energy for protein synthesis and label amino acids so we could identify what proteins they made. I've summarized the experimental procedure on the left, and I've summarized the results on the right. Now, what we discovered was that by far the major product for chloroplast protein synthesis is the large subunit Rubisco. The small subunit, in contrast, is made outside the chloroplast by cytosolic ribosomes, and it's then imported across the chloroplast envelope. So here we have a division of labour, which explains why there are so many ribosomes inside the chloroplast. They make a subunit of the most abundant protein in nature. Now, I've mentioned before that Sam Wyman has discovered that purified Rubisco is one of the very few proteins that will not self-assemble correctly in an amphisin refolding experiment. Because although the Rubisco itself is highly water-soluble, and it occurs at a very high concentration in the chloroplast, 300 grams per litre, the unfolded large subunits are very hydrophobic, and they aggregate together and form non-functional aggregates. So we're surprised to discover that large subunits made inside isolated chloroplasts are actually soluble, even though they're not present in the Rubisco holoenzyme. Now, what we do in this experiment is to isolate chloroplasts from pea leaves, and we incubate them with light as a source of energy for protein synthesis, and we include S35-methionine as a radioactive label so that we can see what proteins the isolated chloroplasts will make. What we do is run the soluble fraction on an undenaturing gel, which simply separates the proteins according to mass. And if you do that, you get this result here. If you look at the stain, which simply stains the bulk protein, you see there's basically two major bands. There are several thousand proteins on this gel, but these two are by far the most abundant. The lower band is Rubisco. That's the Rubisco that the chloroplasts made before we isolated them. Now, if you look at the radioactivity, this is shown on the autoradiograph, you'll see that all the radioactivity migrates with the upper band and not the lower band, especially in the early stages. And if you compare the shape of that stain with the shape of that autoradiograph, you'll see they're identical. They're exactly superimposed. You can see that perhaps most clearly with that and that. It's doing the same thing. Dear me. Now then, if you just think about these results, the conclusion seems rather obvious. Because the stain and the radioactivity coincide, if we can find out what the radioactivity is, we'll know what the stain is, won't we? So what we did was to peptide sequence that radioactivity, and we found it was the large subunit Rubisco. So the conclusion seems obvious, doesn't it? That this protein here, this highly abundant protein, is some oligomeric form of the large subunit that accumulates in the chloroplast before it assembles into the Rubisco holoenzyme. And we published this result in 1970, and since our conclusion seemed so obvious, it was also rather boring, and so we moved on to other things. And I'm ashamed to say that it was not until 1980 that a postdoc called Roger Barraclough discovered that this conclusion, though entirely rational, is also completely wrong. This is the example where Occam's razor led us astray. And the way Roger Barraclough showed this was simply by excising these two bands and running them on an SDS gel, which separates the protein on the basis of their subunits. And then it became clear that this staining band of protein is not the same as the radioactive band of protein, despite the fact that they exactly co-migrate. So let's just excise the 30-minute Rubisco band and run it on an SDS gel and stain it. You've got the large subunit Rubisco and the small subunit Rubisco. There's no radioactivity there at that stage. Now let's take the upper band and do the same thing And then you see that the radioactivity is separate from the stain. That's the radioactive large subunit, but the stain is a different protein. Although they co-migrate exactly, they're not the same. In other words, what we got here is a binding phenomenon. The upper band actually consists of a very small amount of radioactively labeled Rubisco large subunit made in the isolated chloroplast, which is bound non-covalently to a very large amount of another protein which is not made in the chloroplast. It's this binding protein that keeps the subunits from aggregating and allows them later on in the incubation to assemble into the holoenzyme. You can see that label starts to appear later on in the Rubisco. Now, the reason that the radioactivity migrates exactly with the stain is that the stoichiometry of binding is very slow. We know now, in fact, that a single molecule of labeled Rubisco, which has a molecular weight of about 52,000, is bound to a much larger oligomeric protein, which has a size of 800,000. Now, this extreme stoichiometry reflects the complexity of the mechanism by which the binding protein stops Rubisco from aggregating. And I'll discuss what that mechanism is in a moment. Now, we published this result in 1980. We included a time course which showed that the label, the Rubisco large subunit would appear in the Rubisco holoenzyme if you prolong the incubation. We called the binding protein the Rubisco large subunit binding protein and decided to clone and sequence it because it sounded to be novel and interesting. And we made a surprising discovery, which is that it's identical, 50% identical, that means 50% of the amino acid sequence is the same, to a protein from the bacterium Escherichia coli called GROEL. Now, what is GROEL? Well, GROEL was found in the 1970s by several laboratories working on phage replication, the replication of bacteriophages in E. coli. And it was found to be an E. coli protein, which is essential for the bacteriophage to assemble inside the E. coli cell. But why should E. coli make a protein that ensures that it will be killed by the phage? What was its normal function? This was unknown at that time. Now, we interpreted this high identity of two proteins, both implicated in assembly processes, in terms of the molecular chaperone concept. And it was a postdoc, Sean Hemmingson, who actually coined the name chaperonin for this particular family of chaperonins. There are homologs in mitochondria, and there are homologs in the eukaryotic cytosol. This name has caused some confusion, I'm afraid. So remember that the chaperonins are just one particular family amongst over 100 families of other types of chaperone. The chloroplast chaperonin is the most abundant chaperone on the planet because it has to prevent the aggregation of the most abundant protein on the planet. Now, we published this work in a Nature paper in 1988. This is my most cited paper, and this is a picture of Sean Hemmingson as he was then. He returned to Canada, and he runs there the Chaperonin database. Now, it's this paper that sparked a wave of new research in many laboratories in which GROEL and other chaperones are added to amphisim-type refolding experiments. This wave of research actually continues to this day, and it has established, for example, that in the bacterium Escherichia coli, 85 proteins rely absolutely on the chaperonin GROEL to fold correctly inside the cell to avoid aggregation. How they do this, I'll talk about at the end of my talk. Now, I said there were two reports in the 1980s of a newly synthesized protein binding to a pre-existing protein before folding, and the second example was from an animal system. It was found actually with experiments using a cultured cell line derived from lymphocyte precursors. These cells synthesize immunoglobulin, which again is an oligomeric protein made of large chains and light chains. And what Haas and Weibel found was that heavy chains of this immunoglobulin bind to another small protein in the endoplasmic reticulum before they fold. And they call this protein BIP, short for binding protein. and suggested that it was involved somehow in the regulation of immunoglobulin assembly. BIP was later identified by Hugh Pelham and Sean Munro as a member of the heat shock protein 70 family. We now know, of course, that both the chaperonins and the HSP70 chaperones assist the folding of many newly synthesized proteins inside the cell. Now, I referred earlier to the molecular chaperone concept, and because there continues to be widespread misunderstanding about the origin of this idea, I want to point out that the term was not used first by me, but the Cambridge cell biologist Ron Lasky in a Nature paper published in 1978. He used this term to describe the ability of an abundant acidic nuclear protein called nucleoplasmin to solve an aggregation problem during the assembly of nucleosomes in extracts of amphibian eggs. Now, let me remind you about nucleosomes. Nucleosomes consist of oligomers of histone proteins combined with DNA. So the DNA is wrapped around the outside, that's the blue bit, and the histones are on the inside, that's the yellow bit. Now, nucleosomes are stabilized by ionic bonds, because the DNA is acidic and the histones are basic. Now, what Lasky found was that if you break these bonds with high salt, like 1 molar sodium chloride, you can separate the histones from DNA, but if you mix those two together at a sort of physiological salt concentration, which for the nucleus might be 0.1 molar, you do not get nucleosomes. You get an insoluble aggregate, just as you do for Rubisco. But he found that if you add nucleoplasmin first, what that did was bind to the histones, reduce their high charge density, and allow their intrinsic self-assembly properties to predominate over the tendency to aggregate. Now, the term... God, it's doing it again. The term... dear. The term molecular chaperone appears in that paper, and you see what he said about it. And that's the first time the term was used in the literature. Now, why did he choose this term? Well, he chose this term because of the close parallels with the activities of the human chaperone. In both proteins and people, the chaperone does not provide the steric information for the right thing to happen. Nor is the chaperone present in the functional assembled structure. All the chaperone does is to prevent the wrong thing from happening. So the term molecular chaperone, you see, is not a metaphor. It turns out to be a rather precise analogy. Now, notice that nucleoplasmin functions in the late stages of nucleosome assembly, beyond the folding of the histone chains. So the common perception in the literature that chaperones deal exclusively or primarily with protein folding is incorrect. And it has, in fact, been incorrect since this subject started. And in my view, this particular myth has impeded the search for assembly chaperones. Most research on chaperones today still concerns folding chaperones. Now, Lasky did not extend the term molecular chaperone to any other protein or develop the idea into a more general concept, and this is where I entered the story. I noted the functional similarities between nucleoplasmin, the Rubisco binding protein, and BIP, and I made this suggestion in a Nature News and Views article. I put in red here the opening sentence of this article, and you see what I'm suggesting is that maybe chaperones are widespread and involved in both protein folding and assembly in all cells. Now, it's this article that generalized the term molecular chaperone, and rather to my surprise, and certainly to the surprise of Ron Lasky, the term caught on. The graph on the left shows the total number of papers published with the molecular chaperone in the title or abstract. And on the right, I put some of the names. There are at least 100 families of chaperone protein now recognized. I don't know of a comprehensive list, but there's certainly over 100 families of protein which are claimed to be chaperones. So you see what's happened over time is that we've changed our view of protein assembly. Before chaperones, or as I like to think of it, BC, the view was that proteins fold and assemble inside the cell by a self-assembly process that is spontaneous. But after chaperones, we come to a view that proteins use a self-assembly process that is assisted. Now notice that the concept of protein self-assembly itself is unaffected by this change of paradigm. Proteins know how to fold. Their problem is to avoid aggregation inside the cell. Well, all this is history, so let me come to the present. How do we think of this today? And why is aggregation such a universal problem? Well, this slide shows my current definition of a molecular chaperone. It differs from the original definition by including protein unfolding and disassembly as chaperone-assisted processes. Some chaperones, but by no means all, are also stress or heat shock proteins. This is because the requirement for the chaperone function increases if proteins are unfolded by stress, because then they might aggregate. But it's very important to realize that the basic chaperone function is not primarily related to stress. It's required to combat a problem which is created by the very nature of protein synthesis itself. Stress proteins and molecular chaperones, in other words, are not identical sets. They are overlapping sets. And this is another point of definition that's often confused in the literature. There was a recent survey that showed that only 20% of all the chaperone genes in the human genome are actually stress proteins. In other words, 80% of human chaperones are not stress proteins. Now notice that this definition is entirely functional. It's not structural. And it contains no constraints on the mechanisms by which different chaperones may act. This is why I use the imprecise term assist. The term non-covalent is used to exclude those proteins that carry out covalent modifications. Now, I suggest that it's more useful to think in terms of a chaperone function than in terms of thinking of a chaperone molecule. And on this basis, a chaperone can be a property of a protein that has additional and quite different functions. Now, an example of that would be alpha-crystalline, which is a predominant protein found in the lens of the eye. This combines two essential functions in the same molecule. It provides the right transparency and refractive index for vision, but it also carries a chaperone function that combats the loss of transparency as the protein aggregates with age. And when this chaperone function fails, as it usually does, you develop a cataract. Now, this reminds you of my definition of a chaperone function that I gave you at the start. It's the prevention or reversal of incorrect interactions between transiently exposed macromolecular surfaces. Notice that it's not the function of a chaperone to create correct interactions. This is another misleading statement that you see in the literature, statements such as chaperone folds proteins. That's shorthand, but it's misleading. Now, there are at least 6 different types of chaperone function that are currently recognized, subtypes, and I've listed these in the yellow box. Many families prevent or aggregation, and in some cases they also modify the nature of the aggregates so that they're less cytotoxic. There's also a family, the HSP104 family, that functions to dissolve insoluble aggregates. But curiously, this particular family is absent from the human brain. Chaperones are also involved in stabilizing proteins, involved in signal transduction, deciding when a given protein should be degraded, and in preventing premature folding during protein transport across membranes. The last 2 suggested functions are still under intense debate, as I've indicated by the question marks. Now, if you want to hear about the latest research in chaperonology, what better way than to join the UK Molecular Chaperone Club? I founded this club at Warwick some 20 years ago, but it's now run by Pete Lund at Birmingham University. It's got about 120 members. It holds informal discussion meetings every December, and the next meeting will be at Birkbeck College, organised by Helen Sable. I've put Pete Lund's email address on this slide for those interested in joining. Pete Lund is also in the audience, and I'd like to thank him publicly for taking on this task. Now, what I want to finish by talking about is why it is that protein aggregation is such a universal problem inside the cell, despite the fact that most proteins will refold correctly in an Anderson refolding experiment. Well, I think there are 2 reasons. The first is that protein synthesis in vivo takes place on polysomes. And this inevitably brings identical, partly folded protein chains within touching distance of one another. This provides the ideal circumstance for aggregation, because remember that aggregation is highly specific. Chains will only aggregate if they're identical or very similar. What you see on this slide is a polysome spread out on the grid of an electron microscope. So the ribosome is getting on the messenger at this stage and moving along it. We loop out of shot and then back into shot, and you can actually see the extending polypeptide chains coming off, and you'll see that they are within touching distance. The second reason why protein aggregation is universal is because protein synthesis takes place in compartments which are highly crowded with macromolecules. Now, this image is not a section. It's actually a reconstructed three-dimensional image of part of the cytoplasm of the slime mold Dictyostelium, published by Biomaster's lab some years ago. These orange bits are actin filaments, and the grey bits are various macromolecular complexes. And I show you this slide to illustrate the crowded nature of the cellular interior. We use the term crowded rather than concentrated because no single macromolecular species occurs at a high concentration. But if you take them all together, the molecular concentration is a significant fraction of the total volume of the cell. It ranges from about 8% to 40% of the total volume of a cell is physically occupied by the volume of the macromolecules. Now, why is that important? Well, of course, two molecules cannot be in the same place at the same time. So this high degree of crowding effectively increases the concentration of everything. It doesn't increase it by 8 to 40%, which you might think it should. This is why it's so counterintuitive. Crowding theory, in fact, predicts that an 8 to 40% crowding actually increases the effective concentration by between 1 and 3 orders of magnitude, from 10 to 1,000 times. This means that all association reactions, of which protein aggregation is one, of course, will be increased by these very large factors inside the cell. Now, these dramatic effects of crowding were known for a long time, at least 30 years, so it is to be regretted that the vast majority of studies on isolated macromolecules continue to be made in uncrowded media. And this means that the quantitative values of association constants derived from such studies underestimate those which actually operate in the cell. Moreover, this neglect is not necessary because it is possible within limits to mimic crowding in the test tube. What you do is add high concentrations of suitable polymers, such as proteins, which act as crowding agents. And I give one example. The protein alpha-synuclein is the protein that forms the toxic fibers in Parkinson's disease. Now, if you clone this molecule in E. coli, you can get it out in a soluble form, and if you put it in a simple buffer and just incubate it, it will aggregate, but very slowly. It takes days before it aggregates. But if you crowd the solution with various proteins, you see that the rate of aggregation speeds up enormously. In other words, crowding stimulates protein aggregation. So in my view, the prime requirement for the chaperone function is not to do with stress. It's to do with the intrinsic nature of how proteins are made in all cells. The fact that proteins are made on polysomes inside crowded compartments. Now, I'll finish by talking about how some chaperones prevent protein aggregation. I don't have time to talk about assembly chaperones. I'll talk about folding chaperones, and it's useful to divide these into two classes. There's what I call small chaperones, less than 200 kDa, and this includes things like HSP70, HSP40, also membrane chaperones. There are a number of chaperones which occur inside membranes which prevent the aggregation of membrane proteins. Now, how do they work? Well, basically they work by binding transiently to short hydrophobic sequences as they appear on the newly synthesized chain. And they bounce on and off them. They cover up the surfaces for a time. That time is set by slow ATPase activity. So basically what they do is reduce the time the surfaces are exposed. And an analogy has been put forward by Warwick. He says we can call this the hot potato principle. It's like tossing a hot potato from hand to hand until it's cooled sufficiently to hold. So that's a very simple mechanism. Now, if you go to the large chaperone, you get a much more sophisticated mechanism. I call this the Anderson cage mechanism, and it was worked out in a number of laboratories, and I'd like to mention in particular the laboratory of Helen Sable at Birkbeck, the laboratory of Ulrich Hartl at the Max Planck Institute in Munich, and the laboratory of Art Horwitz in the Yale University. What their studies established was that these chaperones assist the folding of newly synthesized proteins by encapsulating each chain one at a time inside a cage. It's a molecular nanocage provided by cavities inside GROEL. I call these amphisin cages for obvious reasons. The cage is actually capped by a removable lid, which is provided by a smaller oligomer called GROES. So if you just look at this section through the chaperoning complex, there are two actual rings, and there's a cavity there and there's a cavity there. This is GROES sitting on one end, and which closes the cavity. GROES can sit on either end. Now, this cartoon shows how we think this works. A partly folded chain coming off the ribosome binds to the open end, and it's the low stoichiometry of this complex that misled us into an erroneous interpretation in the early work on Rubisco. What happens then is that GROES binds to that end, and this triggers a very large change in shape, which extends the size of the chamber. The protein is released into the cage, and now it can fold just as it would in an amphisin refolding experiment, but of course it's got no competing chain to aggregate with. It's been isolated. It's got 10 or 15 seconds to do that, set by slow ATPase, and then the lid comes off and it comes out. Now, it could be that the protein hasn't quite finished folding, in which case it binds back to the same ring and it goes through the same procedure. Now, the sheer ingenuity of this mechanism, I think, illustrates how important it is that the chaperonins, the proteins, must avoid aggregation. Now, I started this talk on an historical note, so I'll finish on one. In the early 1990s, it was suggested that the discovery of molecular chaperones falsifies the principle of protein self-assembly. Now, I hope I've persuaded you that this is not the case, but I decided at that time to write to Amundsen to see what he thought about chaperones. Now, we exchanged several letters, and on the left I put the last letter he wrote a few months before he died in '94. It's not very legible, so I've typed it out here, and you can read what it says. Now, I treasure this letter because in it, obviously, Amundsen clearly welcomed the chaperone concept. Thank you for your attention.
Thank you very much for that. And now we have time for a few questions. The form here is you put your hand up if you want to ask a question. We have two microphones. We will send the microphone to you, one on each side of the room. And then if you could then stand and ask your question, please. So who'd like to ask the first question? Yes, one there. And the next one, so we get them lined up? Yes, at the back. Thank you very much.
Good evening, sir. Thank you for a fascinating lecture. An immediate question which occurs to me is that there may be malfunction of the chaperones themselves, and would that have any bearing on how amyloid, for instance, is made?
I didn't understand the question.
I didn't hear the last bit.
Could you speak up? I can't hear you. I'm sorry. I can't hear you.
Is there any evidence that the chaperones themselves malfunction in your research?
Oh, yes. Well, not in my research, but chaperones are just proteins like all other proteins. They have the same problem of protein aggregation, right? So if you lose the chaperone function, you can't recover it. Now then, if you think about that, it's part of a general problem in biochemistry as to how things get started. Let me give you an example. The function of glycolysis is to make ATP, one function of it, right? But the very first reaction in glycolysis requires ATP. So how does it get started? Well, the answer is, of course, that ATP is always there. Where does it come from? It comes from the previous cell. So the problem goes back to how the cell originated. And there are lots of problems in biochemistry that can only account for when they actually appeared, when the first cell originated, and we don't know how that happened. So once chaperones appear, then it follows. If you lose the chaperone function, you can't recover it. That's why, to me, one of the two really big remaining problems in biology is to explain how cells originated. Do you understand what I'm saying?
Well, the drift of my question was, have you identified any situations where malfunction of these chaperones have led to things like amyloid?
Oh, I understand the question. Yes, I understand. Yes, there are a number of mutations in chaperones themselves that do lead to protein aggregation. I can, if you email me, I can give you the references. Yes, that's certainly the case.
She had the same question, actually.
Same question. Okay. Is there another question? Yes, here. Just wait for the microphone to reach you.
Yeah, thank you very much for your lecture. It's very interesting. I wondered why touching causes folding behavior to change.
Yes, the reason aggregates form is because of a phenomenon called interdigitation. There are particular side chains which, if they're lined up, will interdigitate like that and form a stable structure. And so there are particular sequences with particular side chains which will do that. The ones that are the most productive from that point of view are glutamines and asparagines. If you want to make you can make a peptide which is only 6 residues long, and if it's asparagines and glutamines, if you put them in the right conditions, they will integrate like that. And then that will just multiply up. And so you get a growing fibre. So that's the basic explanation. It's called a steric zipper. And there was a recent survey of what is called the amylone. You can ask the question, how frequently are such aggregation-prone sequences in proteins? And that has been done for the human genome. And the answer basically is that virtually every open reading frame, every gene that codes for a protein, has got at least one of those sequences in it. This is why I say the problem is universal. All proteins in the right conditions will aggregate. So the problem is, as I say, is a universal problem.
Hi, thank you very much for your lecture. I'm particularly interested in how the research you've done is currently or could be applied to medicine. And the reason for that is because I'm the editor of publications, one for pharmacists, and I'd be very interested in publishing something related. So I understand that it's involved in the understanding of Alzheimer's and that there are other conditions that you said you would send in an email to the man across the way. But could you tell me how this is used or may be used in the future for drug treatments for disease?
Yes, you're talking about how the chaperone concept might influence drug treatment in the future. Is that the question?
Well, yes, that's all
Yes. Well, what can I say about that? I can make various points. There is evidence that some chaperones are involved in neurodegenerative diseases. Is that useful at the moment? I would say no, it isn't, because the chaperones that are involved are basically promiscuous. Most work on chaperones concerns proteins which are chaperones which are promiscuous. What I mean by that is that they have a wide range of substrate proteins. So that if you manipulate those, you're going to influence many things. So that's, that doesn't look promising. So what you need to look for are chaperones which are more specific than that. And the work I suggest you follow is the work of Mike Cheetham at UCL. The biggest family of chaperones in humans is the DNAJ family, sometimes called the HSP40 family. There are at least 50 members. And some of these are specific for neurons. And so there is evidence from model systems that if you upregulate those in model systems in cell culture, you can reduce the toxicity of amyloid. So that would be a route I would go for. So that's the first point. The second point I would say is, to my mind, the most important thing in Alzheimer's disease is not actually finding compounds that are effective, because there are lots of studies aiming to do that around the world. Lots of labs are doing that sort of thing, and there are some compounds in various trials. But the problem is that we can't diagnose the disease early enough. The problem is a prodromal analysis. You need a preclinical analysis. You really need to diagnose Alzheimer's 10 years before there are symptoms. How are you going to do that? Well, there are a number of studies around the world trying to identify biomarkers which might indicate that you're going to get Alzheimer's. The most interesting, actually, is in Colombia in South America, where there are some families which suffer from early onset Alzheimer's due to mutation in the appropriate protein. So what's happening is that an American group has set up shop there, and they are monitoring these people as they reach the age of 30, and monitoring them for, sampling the plasma, cerebrospinal fluid, and so on, to see if they can identify any biomarkers which will indicate that you're going to get Alzheimer's. So really, the problem, to my mind, with Alzheimer's is really early diagnosis, long before the symptoms.
Okay, I think this is the last question because we're out of time. Okay. Yes?
Hello. I had a comment and a question. So first of all, the comment is we're just doing some work with Mike Cheetham and show that one of the DNAJ chaperones is protective in a mouse model of Huntington's disease. So I thought your prediction was very good. But my question was that, so aggregates, the mechanism by which aggregates are toxic to cells is debated, and I just wondered what your opinion on those mechanisms might be.
Well, there are at least three different types of mechanism which are not suggested, which are not mutually exclusive. I don't have any particular feeling. I mean, one suggestion is that they interfere with the clearance mechanisms which destroy misfolded proteins. Another is that they interact with membrane proteins. Another is that they interact with various signal molecules. And these could all be correct. So I don't think there's going to be one simple answer to that question.
Okay. All right. So brings us to the end of the lecture and the questions, because we have to be out at the appointed time. I'd like to thank John on all your behalves for a very entertaining lecture. I think you now know all you ever wanted to know about chaperones and more from the horse's mouth, as it were. I think the technical glitches didn't matter at all. We just kept going throughout those. So thank you very much indeed for that. It's a very worthy Croonian lecture. And now this is the nice bit. I have to present John with the scroll with the certificate.
Thank you.
Okay, so shake your hand for
Thank
that.
you.
Okay. A very nice Croonian medal.
Thank you. Thank you.
And a cheque.
Oh, right.
I'm sure that was great.
Thank you.